Let's embark on part 3 of chapter 23, the digestive system, from our textbook of human anatomy and physiology. We're going to start out this time by looking at the regulation of gastric activity. We learned a little bit about the secretions of the stomach during digestion and what they contribute to the digestion. Now we're going to see how it's regulated. So we have all this secretion, up to 3 liters a day of so-called gastric juice, or secretions coming into the GI tract to help digest the food we eat. Take in solids, we've got to add a lot of water to it and enzymes and then break it down into liquid form. There's neural regulation. There are local reflexes, as we said, that control aspects of the stomach function in response to sensory neurons in the stomach. And then we also have long reflexes in which we have activation of digestion by the parasympathetic nervous system, increasing secretion, and motility, it's called. Motility just refers to the smooth muscle activity in the muscularis externa. And then sympathetic neurons inhibit secretion and motility. Hormonal control of the stomach. Well, first of all, gastrin, produced by the stomach, as we saw, by the enteroendocrine cells, we're going to find out that's an activator of enzyme and hydrochloric acid secretion. Affects the primary signal for HCl secretion into the stomach. But then we're going to see a little bit later that some hormones from the intestines feed back and inhibit the stomach's, not to turn it off, but slow down the secretion and motility of the stomach. And we'll see how that works. Okay, so let's look at the regulation of gastric activity and divide it into 3 phases. The cephalic phase of gastric secretion and motility regulation involves sight, taste, and even just thinking about food, the aroma of food. All of those things are sensory inputs into the brain by cranial nerves, and then that's going to influence activation of digestion through the sympathetic or the parasympathetic nervous system. So things start happening. Secretion starts to take place. You may even have felt this surge in your stomach sometimes when you're really, really hungry, and you suddenly get a strong smell of some food that you particularly like. A surge of acid will go right into your stomach of juice being secreted. All right, gastric phase. Once material starts to actually enter in the stomach and distend it, that means stretch it, mechanoreceptors sense that stretch. Peptides in the food that's being ingested, as well as a increase in the pH of the stomach fluid. You add a bunch of solid material and neutral material into the stomach, which is normally very acidic. There's chemoreceptors that can sense that increase in pH. So all those things trigger increased secretion. Got gastrin written on the end of this list because it's a little different from the other things in that list, but gastrin released by the stomach glands will crank up HCl secretion big time. All right, excuse me. What turns on those enteroendocrine cells? all those sensory neurons, as well as peptides and gastrin. So chemoreceptors, that sense changes in pH and the presence of peptides trigger gastrin release, which in turn causes increase in secretion of hydrochloric acid. Okay, so we get things really cranking, start digesting stuff in the stomach and passing some stuff down into the duodenum. As soon as any appreciable amount of chyme goes down into the duodenum, it starts to take over control of the pace of digestion. The duodenum is going to now be the driver from here on out. It's going to feed back and inhibit the stomach activity. That's called the intestinal phase. So in this case, we have 2 things happening. When chyme starts to go down into the duodenum, we're going to have chemoreceptors in there and mechanoreceptors that feed back and inhibit, by way of the enteric plexus, feedback and inhibit stomach activity. That's called the Enterogastric Reflex. Entero means intestine, gastric, stomach. So we have the enterogastric reflex. Once the duodenum starts receiving chyme, it feeds back and inhibits the stomach. Also, the duodenum starts to secrete some hormones from enteroendocrine cells. Only these hormones are different ones, and they feed back and also inhibit. They block the action of gastrins. They inhibit secretion in the intestinal phase. This is the inhibitory phase of stomach regulation. So again, the enterogastric reflex is going to feed back and inhibit the activity of the stomach, the stomach secretion motility. It's actually going to feed all the way back and inhibit vagal nuclei so that we're not getting as much signal to activate stomach function from the parasympathetic nervous system. Is that interesting? It also activates some sympathetic fibers, which will, again, also inhibit gastric function. We're not going to shut it off. We're just going to allow the small intestine to take over the pace, because you see the small intestine is where all the digestion and absorption happens, and we need to keep that going at a rate that can actually lead to the successful absorption of all of the important nutrients, and not let the stomach just get out of control and start pouring stuff in too much. The hormones produced by the duodenum are called enterogastrones. They're hormones that come from the small intestine entero that inhibit the stomach gastrone, and then hormones that owns parts. So enterogastrones include secretin, cholecystokinin, and vasoactive intestinal peptide, VIP. Don't you love that? So those hormones feed back. They go into the blood from the duodenum and feed back and inhibit both the parasympathetic nuclei as well as the actual functioning in the stomach, slow down the stomach motility and so forth. They actually inhibit gastrin activity in the stomach as a hormone. Here's an interesting thing. If the stomach pushes too much stuff into the duodenum, more than it can handle, in fact, especially if there's a lot of solid material, usually mainly what goes down into the duodenum is stuff that's been liquefied in the stomach. Now if we get some significant amount of solids in there, we get what's called dumping syndrome. A very uncomfortable situation. It can involve nausea and vomiting. Here's what happens. As the material goes down into the duodenum, it just draws a vast amount of water by osmosis into the duodenum, which so aggressively that it can actually cause the blood pressure to drop right down and cause fainting. But it causes a lot of discomfort and nausea, and may even cause vomiting. If there's already too much going in the duodenum, your body might say, hey, you got to get rid of the rest of the stuff in the stomach before even more goes down in the duodenum. People that have bariatric surgery, it's a surgery for removing part of the stomach or sealing off part of the stomach, depending on which version of the surgery you have, so that you have less gastric volume. You can't eat as much, and hence you hopefully will lose weight. So people that have this very small, reduced volume stomach, if they have to be very careful how much they eat. They can't just consume a lot of food like us, like people that have a normal GI tract. And if they overdo, they may wind up with getting dumping syndrome. Here's another way of saying all those same things about the phases of gastric regulation. So I don't want to spend a ton of time because it's the same thing, but I'd like you to be able to look at this drawing and gain some value from it, this schematic diagram. So the cephalic phase of digestion involves smelling, seeing, thinking about, and tasting food, and all of those things start to activate digestion right off the bat. Then we come to the gastric phase when stuff actually goes into your stomach. Mechanoreceptors, chemoreceptors that sense increased pH, ones that sense lipid and protein in your stomach contents, start to activate the whole gastric process of secretion and motility. They cause gastrin to be released, and gastrin activates HCl secretion. So we have all these positive factors activating stomach activity, motility, and secretion. And then as chyme starts to go down into the duodenum through this sphincter right here, we're going to have hormones coming from the duodenum that feed back and inhibit, right? We're over on the inhibitory side now, right? All this column is inhibitory on the right, stimulatory on the left. These hormones, these enterogastrones, then feed back and inhibit the secretion and motility of the stomach. and also the enteric reflex, enterogastric reflex. Those sensory receptors in the duodenum will activate neurons in the enteric nervous system. This the plexi in the presence of the wall of the small intestine and the stomach feedback and inhibit the activity of the stomach glands and muscles. All right. Well, your stomach has what's called single unit smooth muscle. What that means is the wall of the organ contains smooth muscle cells linked by gap junctions, and pacemaker cells initiate waves of contraction that spread from cell to cell right through your stomach, passing from the esophagus, from near the esophagus down towards the duodenum, and these happen on a periodic basis. If you start eating food, then we just ramp up the strength of those waves of contraction and secretion by this parasympathetic nervous system. So, distension and the hormone gastrin, in the presence of all the materials in the stomach, start cranking up the muscular activity. So here's a picture of a wave of contraction starting to pass from near the fundus on down through the stomach. And you can see this sort of pinching of the wall, and that pinched in area spreads on down, and it gets tighter and tighter until we reach the pyloric sphincter. All the while, as this wave of contraction is going along, material trapped down closer towards the pyloric sphincter shoots back through this opening between the pinched walls of the stomach, and gets a lot of tremendous mixing going on. And just as the wave of contraction reaches the pyloric sphincter, it relaxes momentarily, and a little bit of chyme goes down into the duodenum and begins that inhibitory process of the stomach that we talked about. So it's a little review. We add materials into the stomach, and things get hypertonic, right? We've got a lot of solids. You add a little bit of water, and you get a very high concentration of solutes in water, hypertonic solution. We have fat coming in and chyme down into the duodenum. It activates, this is the intestinal phase of digestion now. We activate the enteroendocrine cells that are these enterogastrones that inhibit stomach motility and secretion. The chemoreceptors and mechanoreceptors also activate nervous reflexes in the enteric neurons, causing inhibition of the stomach, as well as inhibiting the parasympathetic outflow to the digestive tract and activating sympathetic neurons to slow down the stomach so that the duodenum can take over control of pacing. Emesis is the proper medical term for vomiting. Some of the things that can cause emesis include overstretching the wall of the stomach. If you put too much stuff in there, it'll just send it back. An old parlor game or challenge, I don't know if this still ever exists, that young boys will usually stand up to virtually any challenge they're presented, and a challenge used to be, I bet you can't drink a gallon of milk. And a young boy who really likes milk, especially in the summertime, will think, oh, I can drink a gallon of milk. And so they start to pour it in and pour it in, and pretty soon, once you get to a certain point of stretching that stomach wall, you cannot hold a gallon of milk and you will vomit. Just silly things that happen, but it illustrates this point. Irritants that come into the stomach. Too much alcohol, certain drugs or spices can cause vomiting. Bacterial toxins, when you get, or viruses, certain viruses and bacterial infections in your body can cause you to vomit repeatedly. Cholera bacteria, for example. So there's a vomiting center and a emetic center in your medulla, which receives the signals coming from the stomach that things are not okay down there. Boom, you get rid of the contents of the stomach. The thing is, we've just invested a lot of saliva and a lot of gastric juices. Those are volumes of water that came from the blood, and now we're going to dump them out of the body. We're at risk for dehydration. We've got to replace that water very quickly, or we're going to become dehydrated. We're also going to lose all the electrolytes out of the stomach fluid. And interestingly, if you think back to how the parietal cells made the stomach acid in the first place, if you vomit out all of the acid in the stomach, you're left with an imbalance, because now in the blood is all the bicarbonate ion, the excess bicarbonate ion that was produced during manufacturing of those hydrogen ions. Now you're stuck with them. You're going to have a metabolic alkalosis. Okay, so that's the story about the stomach. Let's move down to talk about the small intestine. The three sections are duodenum, jejunum, and ileum. The duodenum, as you know, is retroperitoneal, the first part of the small intestine, and here we can see it kind of hooks around what's called the head of the pancreas, and then we have these other two parts, the jejunum and ileum, other parts of the small intestine. So the duodenum is a very short part, only about a foot long. It's retroperitoneal. It curves around the head of the pancreas, and it's the place where the pancreatic duct and the biliary duct, the bile duct from the liver, coalesce to form the hepatopancreatic ampulla, and there's a sphincter in there that will allow those two different secretory products to go into the ampulla, and there's another sphincter that allows those things to go into those components to go into the duodenum. So here you can see the liver makes something that's called bile. We'll learn about that later. Stores it in the gallbladder, which is a muscular organ that can then pump that bile through the bile duct down to this hepatopancreatic ampulla. Excuse me. The pancreas produces pancreatic juice, and we'll learn about the composition of that in a bit. And when activated by, as we'll see, some enterogastrones, and the parasympathetic nervous system, we're going to have pancreatic juice formed, and they're going to be released into the duodenum. So this is where we add some important components or aids in digestion from our accessory organs. So then the jejunum is a longer segment, about 2.5 meters long, and then the ileum is the longest part, 3.6 meters. So it takes a long time and a lot of surface area to digest all of the food you eat into its monomeric subunits and then absorb all of those nutrients into your blood. Let's look at 3 adaptations that improve the surface area so that we can get all that done. There are 3 things that the small intestine has that increase the surface area over what it would be if it was just a smooth, glistening, shiny pipe. There are circularly arranged folds in the wall of the small intestine, and we can look at this section right here and see how these folds kind of wrinkle up in the wall. It just creates more surface area. As you're going up and down these mountains, these folds, that is just a lot more surface area than if it was flat. Well, these circular folds are completely covered by villi, little finger-like projections that stick out into the lumen of the intestine, and those are completely covered by simple columnar epithelial cells. Lots of surface area because of, look at all those villi, just millions of them. And then on the surface of the epithelial cells that line those villi are microvilli, the so-called brush border of the epithelium. And so each one of these villi has microvilli on the surface of all these epithelial cells. These are these simple columnar epithelial cells that line the entire GI tract. Inside these, I want you to notice, this is a great cartoon showing the lymphatic system has blind-ended capillaries in the small intestine that are unique. They're specialized. They're called lymphatic lacteals, and as we're going to see later, that's the site of absorption of lipids from the diet. And then we have lots of blood capillaries in these villi, the site of absorption of sugars and amino acids. We also notice something about the anatomy here in the mucosa of the small intestine. There are lots of these so-called intestinal crypts, and there's gland cells in there that can help make some secretions that are important for the digestive process. There's also some stem cells in there, some immortal cells that can replace all of these epithelial cells on a regular basis. And there's a little micrograph of a villus, and you can see all those simple columnar epithelial cells as well as some of the blood vessels that are in that villus. Here is a beautiful micrograph of microvilli. The microvilli, the brush border on the surface of a simple columnar epithelial cell, serves to increase its surface area by about 300 times. That's quite impressive. So we have 3 important adaptations to help us have enough surface area to absorb all of the nutrients from our diet. All right, join me next time for part 4, which we'll start talking about the accessory organs of the liver and the pancreas, as well as the functioning of the enterogastrones. We already know the enterogastrones feedback and inhibit gastric motility and secretion. Well, they're going to actually activate. They're the things that regulate, to some degree, the accessory organs.